Okay, so this is my final video in the series Evolutionists Say the Darndest Things, and I'm just going to use this video to go through a quick series of comments that people have said to me when I was having arguments with them about evolution. These people are all highly educated and they've got various qualifications, and they strongly believe in the evolution theory, strongly believe it enough to engage with me in conversation. So let's have a look into some of the things that they said. I've broken it down into smaller chunks so that it's easier to break the information down. So the first part that we'll be dealing with is that DNA doesn't actually change. Never changes, there's no evidence of that. So I had an argument and this guy got involved and he said DNA is not unchangeable, so I must have said that DNA is unchangeable, and his response to that was DNA is not unchangeable, you have around 200 to 300 mutations from your parents. Offspring are different from their parents. You have 200 mutations from your parents over many generations. That is how a new species comes about. And then, when he was getting more exasperated, he said, all organisms have different offspring. You have around 300 mutations different from either parent. Now, it's interesting that he wants to use mutations as the evidence that evolution over long periods of time does happen, because the overwhelming evidence that we have of mutations is that overwhelmingly they are actually harmful to the functioning of a species. So let's look into that now. So a mutation can result in many different types of change in sequences. Mutations in genes can either have no effect, alter the product of a gene, or prevent the gene from functioning properly or completely. Mutations can also occur in non-genic regions. One study on genetic variations between different species of Drosophila suggests that if a mutation changes a protein produced by a gene, the result is likely to be harmful, with an estimated 70% of amino acid polymorphisms that have damaging effects, and the remainder being either neutral or marginally beneficial. So he's using mutation as the key evidence for an example that evolution does happen. But as we're looking into it here now, mutations actually affect the protein that is produced by a gene, and in an estimated 70% of the time it damages the functioning of the gene. In the other cases, it's either neutral, it has no effect, or it's marginally beneficial. So that's not a process that's going to bring about a completely entirely new species. If 70% of the things that are happening to the functioning of the genes is harmful, then you're actually regressing the gene code, you're not forwarding it. Due to the damaging effects that mutations can have on genes, organisms have mechanisms such as DNA repair to prevent or correct mutations by reverting the mutated sequence back to its original state. So get another wee quick definition. Mutations. Changes to genes are called mutations. Mutations can be spontaneous, they just happen. They can also happen because of radiation or chemicals such as tar from cigarette smoke. Mutations may have no effect. For example, the protein that a mutated gene produces may work just as well as the protein from the non-mutated gene. Mutations may sometimes be helpful, but they are often harmful. For example, haemophilia is an inherited disorder that stops blood from clotting properly. It is caused by a mutated gene. So it's interesting that evolutionists would want you to believe that mutations can lead to a completely entirely new species being born, and that is the best example that they can give, that genes can change, that DNA can change, but it doesn't actually change the DNA, but it changes the way that the proteins created by the DNA works. And another thing is, when they go to give examples of mutations, all that they can give is examples of things harming the functioning of the genes, the functioning of the species. So the examples that they've got of mutations happening here in this slide is that you can get mutations happening if you suffer a bout of radiation, or you suffer a bout of some form of chemical attack. So that's not a process that's going to bring about an entirely new species. That is going to be a process that brings about the destruction of the species. Now, another thing is that DNA doesn't actually change. What happens is that DNA is actually encoded by a thing called RNA, and RNA is actually far more important than the actual gene code. The RNA is what happens, how the species actually functions. The DNA is like the motherboard, the computer circuitry of the genome, but the RNA is the part that decides what actual genes are switching on and switching off, what genes are going to be used in the actual functioning of this being. So genes make up only 2% of the human genome and were easy to spot, but the on-off switches controlling these genes were encrypted within the remaining 98% of the genome. Without these switches, called regulatory DNA, that's the RNA, genes are inert. So the DNA is there, the RNA can then select which parts of the DNA it wants to use and turn on, and that is the functioning of the species. The DNA can't actually change, but the RNA can select parts. It seems like the DNA can change, but it actually can't, but the RNA is just selecting different parts. Now, there's a thing called the central dogma of biology, and what that is I'll just kind of go through it quickly to explain the functioning of DNA and RNA. So what is the central dogma? The central dogma is the process by which the instructions in DNA are converted into a functional product. It was first proposed in 1958 by Francis Crick, discoverer of the structure of DNA. The central dogma of molecular biology explains the flow of genetic information from DNA to RNA to make a functional product, a protein. And this is the same across the board for every single example of any living organism. So RNA is ribonucleic acid, a nucleic acid present in all living cells. Its principal role is to act as a messenger carrying instructions from DNA for controlling the synthesis of proteins, although in some viruses, RNA rather than DNA carries the genetic information. So DNA is deoxyribonucleic acid, a self-replicating material which is present in nearly all living organisms as the main constituent of chromosomes. It is the carrier of genetic information. So the DNA has all the genetic information to select from, but the RNA actually selects the information in use for the species. The central dogma suggests that DNA contains the information needed to make all of our proteins, and that RNA is a messenger that carries this information to the ribosomes. The ribosomes serve as factories in the cell where the information is translated from a code into the functional product. So the DNA is the code, and the RNA selects the switches from the code to turn on. The process by which the DNA instructions are converted into the functional product is called gene expression. Gene expression has two stages, a transcription and a translation. In transcription, the information in the DNA of every cell is converted into small, portable RNA messages. During translation, these messages travel from where the DNA is in the cell nucleus to the ribosomes, where they are read to make specific proteins. The central dogma states that the pattern of information that occurs most frequently in our cells is from existing DNA to make new DNA. So the DNA exists, the RNA can replicate it. From DNA to make new RNA, that's transcription, so the RNA can make new RNA, transcription, and from RNA to make new proteins, and that's translation. So DNA can't actually do anything, the RNA actually does it all, but the DNA can't actually change. So the RNA can make new DNA, it can transcript to make new RNA, and it can also translate to make new proteins. Reverse transcription is the transfer of information from RNA to make new DNA. This occurs in the case of retroviruses such as HIV. It is the process by which the genetic information from RNA is assembled into new DNA. So it's interesting there, I guess that they are maybe then trying to say that from that process DNA can change, but the only example that they have of that is HIV, and HIV is probably the worst chronic illness that man has ever occurred. So to say that, okay, so you can change DNA, and the example that we've got of that is HIV, that's not really an example of evolution, is it? That's an example of the complete destruction of the DNA. So First of all, natural selection can't actually change anything. Natural selection is exactly the process that we've just went through. DNA doesn't change. What happens is natural selection is basically talking about the RNA. The RNA can select various parts of the genome to express, but natural selection has no creative properties. It can't create a new being, it can't create new DNA, it can only select from what is in the actual DNA code of that particular species, and natural selection has its limits. Because natural selection can produce amazing adaptations, it's tempting to think of it as an all-powerful force urging organisms on, constantly pushing them in the direction of progress, but this is not what natural selection is like at all. First, natural selection is not all-powerful, it does not produce perfection. If your genes are good enough, you'll get some offspring into the next generation. You don't have to be perfect. This should be pretty clear just by looking at the populations around us. People may have genes for genetic diseases. Plants may not have the genes to survive a drought. A predator may not be quite fast enough to catch her prey every time she is hungry. No population or organism is perfectly adapted. Second, it's more accurate to think of natural selection as a process rather than as a guiding hand. Natural selection is the simple result of variation, differential reproduction, and heredity. It is mindless and mechanistic. It has no goals. It's not striving to produce progress or a balanced ecosystem. So that's just basically what I'm saying. Natural selection has no creative properties. From the dabblings that you've had in evolution, you might think that natural selection is the cornerstone of evolution and how species change and move forward, but it's actually not. So it's not actually survival of the fittest, it's survival of the fit enough. There are many reasons why natural selection may not produce a perfectly engineered trait. For example, you might imagine that cheetahs would be more fit, produce more offspring if they could run just a little faster and catch more prey. Here are a few reasons why natural selection might not produce perfection or faster cheetahs. It's a lack of necessary genetic variation. Selection can only operate on the available genetic variation. It can only operate on what is already in the DNA code in the genome of that species. It can't push it beyond what the species is capable of. A cheetah might run faster if it had faster genes, but if faster genes are not in the population from mutation or gene flow evolution, in this direction will not happen. A faster cheetah might evolve if the fastest cheetah in this population passed its faster genes to its offspring. Constraints due to history. Perhaps a different arrangement of leg muscles and bones would produce cheetahs that run faster. However, the basic body form of mammals is already laid out in their genes and development in such a mutually constrained way that it is unlikely to be altered. There really may be no way to get from there to here. So I guess that's just an explanation that fundamentally you can't go beyond the limits of what your species is. And that's not an example of evolution at all, but evolution, if you dabbled in evolution, it would have you believe that natural selection is the cornerstone of how species change and move forward. Trade-offs. Changing one feature for the better might change another for the worse. Perhaps faster genes are in the population, but there is a trade-off associated with them. Running faster for short distances means the cheetah's metabolism requires even more energy, or that the cheetah's legs must become hazardously delicate. Although longer limb bones increase stride, their chances of failing due to bending loads increases as well. In this case, perhaps it would get no net increase in fitness as a result of the faster genes. So maybe if the cheetah's tibia and fibia, whatever it might be, were a bit longer, it could actually run faster, but if the bones were longer, they would probably be thinner, more brittle, break easier. So with every single thing that might possibly change, then there's also a blowback from that. And at the end of the day, you can't actually go beyond what is in the gene code, what is already in the DNA. If the DNA is specific to have bones at a certain thickness and a certain length, it's never really going to go beyond that. And even if it were to go beyond that, it would have a negative impact as well as a positive impact. It's not a process that is going to change lasting evolution. So the interesting thing is that any part of the RNA is selecting things in the DNA, but the DNA can't physically change it. It can't actually be altered. And the evidence that we have for that is that there was bacteria found in sailors who had frozen in the ice long ago, and they pulled their bodies out and they found that there was bacteria viruses around them, and those bacteria were resistant to penicillin before penicillin was actually invented. So what that means is that the ability to be resistant towards penicillin was already in the DNA, was already in the gene code before it had the need to be selected. So everything that is actually in the gene code is solidified, doesn't change, can't change. The RNA can select various parts of the gene code and turn it on, but this example of the bacteria being resistant to penicillin before penicillin was invented is an example that everything that is in your gene code that can possibly be selected from is already there. It's been there since the beginning of time, and it will be there till the end of time. Everything that's in your gene code is actually already there, and all the RNA is doing is selecting it. So bacteria from long dead sailors proves resistant to antibiotics. So frozen corpses of 2 seamen who died during a doomed Arctic expedition in 1848 contain bacteria that show unexpected resistance to antibiotics developed more than 120 years later, scientists say. The bacteria came from the bodies of William Brain and John Hartnell, 2 of the 129 men who died in the Canadian Arctic during Sir John Franklin's doomed 1845 to 1848 search for a northwest passage connecting the Atlantic and the Pacific. In 1986, the well-preserved bodies were among 3 retrieved from graves in the permafrost on Beechey Island, west of Baffin Bay in Canada's Northwest Territories, by Canadian anthropologist Owen Beattie, co-founder of the 1987 book Frozen in Time: Unlocking the Secrets of the Franklin Expedition. 6 types of bacteria lying dormant for over a century were found in the bowel contents of Brain and Hartnell, Gruchkowska said. The organisms apparently survived 140 years in permafrost. They grew sufficiently in the laboratory to be identified and tested for susceptibility to currently used antibiotics. She said 2 of the 6 strains of bacteria, all of which belong to the Clostridium genus or group of species, were found to be partly resistant to the antibiotics cefoxitin, which was introduced in 1978, and clindamycin, introduced in 1970. The surprising discovery may spur revision of the theory that the proliferation of bacteria that survive antibiotics is solely caused by widespread use and abuse of antibiotics that kill vulnerable germs, she said. Mrs. Gruchkowska said tissue specimens from the corpses were kept frozen in sterile containers until bacterial tests were performed, assuring there really were 140-year-old bacteria. So as I said, that is one of the proofs that whatever exists in your DNA code is already there and will always be there. The RNA can select it, but you can't actually go from species to species. So another thing to kind of double down on the information, going back to the central dogma of molecular biology. The dogma is a framework for understanding the transfer of sequence information between information carrying bipolymers in the most common or general case in living organisms. There are 3 major classes of such bipolymers: DNA and RNA, and they are both nucleic acids and protein. So your whole entire DNA is made up of, the function of the DNA is made up of 3 things: DNA, RNA and protein. There are 3 times 3 equals 9 conceivable direct transfers of information that can occur between these. So out of all of the information, out of all of the massive expanse of information that is held in DNA, there are actually only 9 conceivable ways of transferring that information into proteins and into a useful thing. The actual functioning of the animal, the species, the organism, there are only 9 possible ways that DNA and RNA can actually function. So that is mind-blowing, and it's, if anything, it's proof of a god, it's not proof of evolution. The dogma classes these into three groups of three. Three general transfers believed to occur normally in most cells, three special transfers known to occur but only under specific conditions in the case of some viruses or in a laboratory, and three unknown transfers believed never to occur. So that is even more galling then for evolution. Certainly I would say goes towards my belief of God, because you've got DNA and all the information that's held in that is absolutely mind-blowing. But there's only 9 possible ways that they can actually function in the daily operating of a living organism. 3 of them are general transfers that are happening all the time. 3 of them are special transfers that happen a very, very small amount of time. And 3 of them are unknown transfers believed never to occur. So out of the 9 conceivable ways, basically only 3 of them are ever happening at any given time. The general transfers describe the normal flow of biological information. DNA can be copied to DNA, that's replication. DNA can be copied into RNA, that's transcription. And proteins can be synthesized using the information in RNA as a template. So that's all that DNA can do. It can replicate itself, it can make RNA, and RNA can then make proteins. So that's it. That is the central dogma of molecular biology and every single living organism. That's all that DNA and RNA can do. They cannot create completely new species. So another thing to consider is that if evolution really is happening, right, let's look around us and get some information about what's actually happening around us to see how successful this evolution is. Well, unfortunately, there's actually a stunning amount of people who live with disability and a stunning amount of people who live with chronic illness and disability and mutated genes in the negative sense, and live with faulty genes, and we see that in much greater number. Think how many people that you see who have a faulty gene or have some kind of illness compared to the amount of people that you see who are much more intelligent than the normal person, are much stronger than the normal person, are much taller than the normal person. When do you see people like that are not strong within the general range of human being capability, are not intelligent within the general range of human being capability? But we see literally millions of people who live with faulty genes. So the official statistics for the UK, the general demographics, is that there are over 11 million people with a limiting long-term illness, impairment or disability. The most commonly reported impairments are those that affect mobility, lifting or carrying. So the United Kingdom has a population of 65 million, so 65 million to get 1% is 650,000. 650,000 times 17 is 11,050,000, so 17% of the UK population have a limiting long-term illness, impairment or disability. Does that sound to you like it's a process of evolution where we're going towards forming a higher species? No, and it's the same across the board for any organism. So disability prevalence estimates for 2011 and 2012. The Office for Disability Issues has updated Department for Work and Pensions estimates, which shows that there are 11.6 million disabled people in Great Britain, of whom 5.7 million are adults of working age, 5.1 million are over pension age, and 0.8 million are children. This estimate covers the number of people with a long-standing illness, disability or infirmity and who have a significant difficulty with day-to-day activities. So 11.6 million people in Britain have a significant difficulty with day-to-day activities out of 65 million people. That's not a process that is taking us towards evolution. That's a process of people who are born with, unfortunately, in such unfortunate circumstances that they actually find it hard to even function on a daily basis. Now, another thing is that disability doesn't even actually take into account all the faulty genes that are out there. There's plenty of faulty genes that you can have which don't actually affect your functioning on a daily basis, but they would still be detrimental to your ability to survive in the wild. And what evolution is it's the ability to survive in the wild. That's what it is. Evolution is the idea that a species can change just directly through breeding and having a relationship with the environment around them. And that's all it is, just breeding and changing to adapt to the environment. You couldn't change to adapt to the environment if you were disabled. If you were disabled, your species would die out. If you had lots and lots of circumstances where faulty genes keep coming up, then you're going to find it very hard to survive in the wild without technology, etc. So 74% of people in the UK either wear corrective eyewear or have had laser eye surgery to help them see better. More women than men wear corrective eyewear. The use of corrective eyewear in the UK is very common and has not changed significantly from 2011. Of those needing corrective eyewear, the majority wear it to correct short sight. 69% of people in the UK wear glasses. 13% of people in the UK wear contact lenses. So 74% of Britain's population are likely to be short-sighted, or they can't see very well and they need glasses to correct their sight. If glasses weren't invented, how possibly would the human species survive well if most of us couldn't even see to hunt properly? So what percentage of the population wears glasses? According to the Vision Council of America, so this is America now, approximately 75% of adults use some sort of vision correction, about 64% of them wear glasses, and about 11% of them wear contact lenses, either exclusively or with glasses. Over half of all women and about 42% of men wear glasses. Similarly, more women than men, 18% and 14% respectively, wear contacts. Of those who use both contacts and eyeglasses, 62% wear contact lenses more often. So 75% of America have bad eyesight. In Great Britain in 2009, around 1 in 3 adults, 30% of the population, reported that they had a long-standing illness or disability, compared with around 1 in 4 adults in 1972. So as time is going on, we're actually getting more disabled, more dysfunctional, more examples of faulty genes being passed on. So what we're seeing here is that, first of all, do you think that you're really going to be surviving in the wild wearing a thick-ass set of glasses? I don't think so. You're not going to be able to hunt. You're not going to be able to see prey coming in to attack you. You're not going to be able to defend yourself. You're going to be a sitting duck if you can't even see properly, and 75% and 74% of the UK and the USA respectively need glasses to function on a daily basis, and that is very, very galling for the evolution theory, because there's no possible way that people who had bad eyesight would be able to survive in the wild. And if you're saying that 75% of the entire population suffer from this problem, then it's not an example that evolution does happen. It's actually an example that actually more often bad genes are passed on, not a prevalence in a species to become an entirely new species. And as the stats there show, we're actually getting more dysfunctional, our genes are getting more dysfunctional and of poorer quality in general as time goes on. So another thing is that from generation to generation, it doesn't mean that what happens with the DNA is it's a complete scattergun approach. There is nothing lasting whatsoever about DNA and DNA transfer. There's nothing lasting whatsoever about breeding. I don't wear glasses, but my mother wears glasses, so that I have skipped that gene. That gene didn't come to me, even though 75% of the population do need glasses. So with each generation, it's a complete scattergun approach. There is no real evidence of lasting change in any species. It just basically doesn't happen. A way of proving that is to say, well, so Down syndrome, do they always have Down syndrome babies? So the answer is no. At least half of all women with Down syndrome do ovulate and are fertile. Between 35 and 50% of children born to mothers with Down syndrome are likely to have trisomy 21 or other developmental disability. So that's only 35 to 50% of Down syndrome mothers are giving birth to a baby that has developmental disabilities. That goes to prove that what nature is actually, DNA and RNA, the fact that it that it doesn't have lasting change is a good thing, because it means that that is how we survive. We actually survive because DNA doesn't have lasting change. It actually has a complete scattergun approach, and each generation has the same ability to be different from any other generation. So Down syndrome people do not have Down syndrome babies 100% of the time with this approach, with the approach that each generation is different, right? If you have a negative trait, the negative trait can be overridden and it can go back to normal. And unfortunately, it's the same with a positive trait. If you have a positive trait, it can be overridden and go back to normal. Basically, in all circumstances of DNA and all organisms, what it's striving to do is to go back to normal. That's what it's striving to do. It's not striving to create a completely new species. There is no evidence for that. What the DNA is striving to do is to become normal, to become standard. Again, so do dwarf people have dwarf babies? And the answer is no. Two parents with dwarfism can have a child of average height because dwarfism is a dominant trait, and the parents probably did not inherit their dwarfism from their parents. At some point early in development, their FGFR3 gene picked up a DNA change that led to dwarfism. So what's happening is that answer is actually very galling for the evolution theory, because they're saying that dwarfism is a dominant gene, so most people should be small, but they're actually not. So I guess I'll explain it further in later examples, but what that could be is that the RNA is enacting the gene, the dwarf gene, it's switching it off. So even although the dwarf gene is dominant, obviously in most cases of people the RNA must be switching that off, because not most people are small. So it's very galling for the evolution theory though to have a trait that we see as being a disadvantage as the dominant gene. That's what that's saying. But what I'm saying anyway, the point of this is that in all circumstances, do dwarf parents have dwarf babies? And the answer is no. So what we're saying is that the desire for the DNA, the way that the DNA is expressed, it's completely scattergun approach with each ensuing generation, and what happens is that in more circumstances than normal, the desire or the way that the process works is that eventually it will go back to normal, and that is what the process takes us. The process takes us on a path back to normality. It doesn't take us on a path to extreme. So that's an example there. Tiffany Di Donato is a mother who gave birth, a mother suffering from dwarfism who gave birth to a normal sized child. And this is an example of a family where the mother and the father at the front and centre there are suffering from dwarfism, and they have 4 children, and I think, is it 3 of them are normal sized and 1 of them suffers from dwarfism. And that's a television show, Little People, Big World. The show follows the daily lives of the Roloff family, parents Matt and Amy and their 4 children, Zach, Jeremy, Molly and Jacob. Matt, Amy and Zach have dwarfism, while Jeremy, Molly and Jacob are of average height. Zach and Jeremy are fraternal twins, although Jeremy is of average height. His brother Zach is a little person. So what I'm saying there is that not all people who have dysfunctional genes pass those genes on to their children, and it's the same with any kind of positive trait. Also, negative traits aren't necessarily passed on to the next generation, and positive traits aren't necessarily passed on to the next generation. It's a complete scattergun approach for the actual functioning of genes in the real world. So there's Usain Bolt there. He is a special human. He has special capabilities. He's the fastest human that we know of in recorded history. When he has a child, right, do you think that his child will automatically then be the next fastest human in the world or faster than him? And the answer is that very most likely will not be. The person that he gives birth to will very likely not have his special capabilities. So if we look at the evidence around us, they want us to believe that evolution occurred and that in evolution we are all descended from a common ancestor. So every single organism on Earth has came from one organism that came alive however many millions or billions of years ago. But if we look at Usain Bolt, today and realise that he isn't going to give birth to a human who is as special as he is, then where can we possibly get this evidence that generations can pass on their positive traits or their negative traits for that matter? It doesn't happen, and there is no evidence for that. So all of the evidence that I've just shown you there is the culmination that DNA itself doesn't actually change. It can't change. RNA processes the information in the genome. That's the natural selective part of gene expression. The information that's in the genome has existed from the start of time, and it will exist until the end of time, and the RNA within that particular species can be selected in various ways, but it can't actually produce entirely new beings. But DNA itself is unchangeable.